short announcement that this is going to be the last presentation in this session before the break. Our last speaker could not make this session. So, Alexis.
Good, perfect. So I can take his time. Okay. So hi, my name is John Alexis Guerra Gomez. I invite you to take a look at my Twitter. If you guys want to follow the slides, they are available online, and also the code, the demo, the video, etc., etc. So I'm going to be presenting today BioCicle, that it's my students Meili Vanegas Hernandez work when I was working at Los Andes University in Colombia, working together with this amazing group of collaborators. So to give you a quick idea, because we don't have much time for presenting, we are contributing a couple of things in this paper. So the first one is taxonomy, not a phylogenetic or phylogenetic tree or things like that, but a task-focused taxonomy of the different type of activities that you want to do when you're doing sequence alignment in biology. And then, using part of that taxonomy, we show BioCicle that it's a tool that helps you summarizing and comparing to such things. So to start, let's start explaining why we need BioCicle. So BioCicle came from working with biologists that tell us that he's commonly doing sequence extraction. And in things like metagenomics, it's very common that you take those sequences and you want to characterize them. You want to see if I just found a new organism that hasn't been classified, or what are the different organisms that look similar to this one that I already have. So usually when you do that, you compare with tools like BLAST or HMM to different databases that already contain millions of different organisms already known. And the problem is that depending on the size of these databases and the number of errors they have, you can get misclassifications. You can get an organism that has been classified as unknown, for instance, because people don't really know that many details about them. And then if you assume that that's your final result, then you start having the problem of creating error propagation, because I could publish a paper that actually uses those errors. So it's pretty much the best way I like to think about this is like doing a Google search and only looking at the first result. Whereas in here, we wanted to look at all the different pages and summarize that. The problem becomes even harder when you are not comparing only one sequence, but many of those. And in those cases, we needed better tools, or our expert needed better tools for doing this. Usually this is a simplified version of what you get when you perform this type of comparisons. So usually you say, I just sequence this bacteria or virus or something in here, and then I got a set of results out of the system telling me, okay, these are all the different organisms that we already know. These are the names for them, and then you get a score that tells you how close they are. And then you also have like the trees that you have been listening the whole day today. So basically with this, we set up to build a tool called BioCicle. It's web-based, it's open source. You should be able to access it today, so I invite you all to break the server and to crash it. It works with the outputs that you can get out of the NCBI tool just by an accession ID, although in the version that it's online, you don't have that capability. But if we push the developer, maybe she will bring it back. Or it could be also work on pre-compute comparisons that you run on your clouds. It works with single and multiple sequences, so let's get to it. So as I was saying before, when you're doing these comparisons, you get a new sequence, and then you want to see if it is similar to something that has already been discovered. So you get these score values and these taxonomic ranks, as they call them. And what we are going to be using is encoding using the spatial region, the whole taxonomic range, and then using the length, the score value. How does that work? Basically, as I was mentioning before, either you run your BLAST on your cloud, and then you get an XML output, and then you can feed that directly in here. So that's one thing that very differentiates us from, excuse me, from other tools that we saw in the literature, is that we use standard formats. And we even connect with the APIs from NCBI and UniProt for protein accessions IDs. And then once you get that, you get our interactive visualization that you can use for summarizing those things. So in this case, this is one of the views that you can get on the tool. As you can see, it's very crowded. It contains first a summary of all the sequences that you are comparing. Then you have a phylogenetic tree, a small version of the tree that helps you navigate, and a detailed view of one of the comparisons. So that detailed view is something like this. It's a zoomable icicle tree where we are representing using the x-axis, the different characteristics of the tree. And then the height represents the scores. So the advantage of using something like this is that it can be a little bit more readable, even though it uses in a less efficient way the space than radial layout. But once you start zooming in, you can get more readable labels. And then at the same time, it's very easy to aggregate what is the most prominent organism that was a match for this. And this is for one sequence versus all of the elements. For the summary, each one of those that you saw before can become like one of these small thumbnails in here. So you're looking at something like 200 or a little bit more of sequences in there. And then the fact that we're using colors for representing the families, it actually helps you understanding if it is close or far from different type of elements. Then we have a very ugly dendrogram on the left that can be actually very helpful because it helps you filter and search through the different branches to try to see, I only want to see samples that belong to this part of the tree. And then finally, a feature that was requested by our expert is that there is a score threshold that you can use to say, I only want to keep in the tree all of the matches that are above this certain score that it's important to me. So bringing all of that together, let me show you a small case study because we don't have much time. But you can go, as I mentioned before, on my Twitter and start playing with it online. So here is a simple example when I had 179 sequences out of the 16S gene, which I have no idea what it is. And basically, so actually the biologist has this. And then he wanted to know, like, what are the type of organisms that were in there. So when you look at the small multiples, this one in here, you can get, like, if you are somebody from visualization like me and don't understand any of this, then you see a lot of purples, a lot of yellows, and a lot of oranges. So that actually translates very well on the type of organisms that you see in there. So when you actually see in the dendrogram, this is a very small version, but it actually shows you that on the protobacteria, there is 125 of those elements that are divided into gamma and alpha, and then there are bacteroides in there. So the nicest thing is that you can go and select and see out of the 179, I only want to see the proteobacteria. And then out of those, I only want to see the gamma bacteria. And now you start seeing that the colors start like filtering, and you can get a faster idea of these comparisons. The same goes if you go into alpha, or if you go into flava, or whatever that means. So here is just a simple summary of all of those things. So you can get a very quickly navigation and start exploring and try to get an idea on what are the different elements that I just sequenced in there. There is a nice video with a more beautiful voice of my student, but we don't have time for that, so let's go to the second contribution. So the other thing we did is that in order to perform this and to build this, we have found a lot of tools that were built in the state of the art, but we kind of had the idea that they weren't built using like visual analytics principles, or at least not from our side of the pond. So basically what we did is that we say, let's start classifying them and start to see how they fit into a whole taxonomy, because we love trees so much. So the whole idea is that when you have one sequence and you are trying to characterize that and compare that with another sequence, then you can get three types of results. That's what we found. So we found that you can get regions of similarity, you can get taxonomic profiles that we have here a lot of today, or you can even have like sequence descriptions, like something that tells you that thing that you're looking at in there, it's a weird Colombian speaking in front of a crowd, okay? So the other thing you can get is what if you are comparing like that sequence versus a whole bunch of other sequences, that is the database comparisons that we had. So you can still have the same type of results, it's just that you have way more elements in there. So again, regions of similarity, profiles, and then descriptions. So out of those three categories, and remember we are comparing only one sequence now, we can also add, what if I compare multiple sequences? And that brings us this nice table that you see in here. And this is kind of our contribution for this. We took this table, it's a task-centered taxonomy. Then you can see we have comparing one sequence or comparing multiple sequences for the three type of results. And these are all the different projects that we could find in the state of the art, based on the fact that they were comparing either one sequence or multiple sequences and what type of results they were analyzing. On top of that, we also added a small subsample in there that were the tools that actually supported non-restrictive inputs that are the ones that you can find in here. And the one closest to ours is Corona. And actually, BioCicle fits in these two areas. We also support one sequence or multiple sequences. So we also have more information, but I will relate to the paper because I'm running out of time, about the different pros and cons of all of the tools that we found online. We also welcome your feedback, telling us how much you love it or hate it. But just to wrap up, our two main contributions of this work is that taxonomy that we invite you to consider, because maybe in the future we can start placing all of your amazing tools in this, or expanding that, or saying why it works or not. And then one tool that focuses on two of those areas that is called BioCicle, that is open source, and you guys can go ahead and start using right now. And with that, I will welcome any questions that you might have. There is the link for the code, and again, my Twitter, if you guys want to see these on your computers, on your data. Thank you very much. Did I scare you enough?
For the audience who might be biologists, could you explain what visual analytics is?
Oh, that's a tricky question. So my answer, and I'm scared to answer this question now. The visual analytics field is this field where we are trying to measure the value of visualizations, like how you can create interactive interfaces that allow experts to extract insights from data. So in that sense, and piggybacking on Petra's talk, that insight or that value can be just providing emotions, but in most cases, and for the vast majority of you, will be to be able to perform in a more efficient and effective way tasks on different data sets. So basically, it's a tool that allows you to do something that you couldn't do before in a more effective way. That will be I hope I didn't fail.
All right, then just a reminder that this was the last talk of this session, and we will see you all again at 4:40.